Hey guys, and welcome to Young-Splaining Photosynthesis. So, what is photosynthesis? Well, photosynthesis is an energy transferring anabolic process. In a nutshell, the energy is going to be captured from sunlight and stored in the chemical bonds of created organic compounds. So, photo for the light capture and synthesis for the created organic compounds. The energy from the light is going to be stored in the chemical bonds of those compounds. Now, photosynthesis, let's look at it in a plant. Photosynthesis is going to occur in the green parts of the plant. So let's look at the cross section of a leaf. So, one of the first things I want to draw your attention to are these cells down here at the bottom. Now, these cells are the guard cells, and I want you to notice that oxygen is coming out and carbon dioxide is going in. So plants do respire, they do exchange gases. The next cells I want you to notice are the two cells that's outlined. The top layer is the mesophyll layer of cells, and the bottom one is the bundle sheath cells. Now, these cells are where you're going to find the chloroplast. The chloroplast is going to be located in the mesophyll cells and in the bundle sheath cells. That purple thing next to it is what I like to call the vein of the plant. And what's actually making that up are the xylem and the phloem. If you ever look at a leaf and you see like the little veins in the leaf, you have the xylem and the phloem. And these are special transport tissues. They function in transport. Xy goes high, so it carries water from the root to the leaves of the plant, and flow goes low, so it's going to carry those organic sugars that are made in photosynthesis throughout the rest of the plant. Some of it will be stored, some of it will be incorporated into other parts the plant needs, and some of it will be used. Now, back to this guard cell. This guard cell is called a stoma, and I like to think of the stoma as being the nose of the cell because this guard cell does open and close. And by opening and closing, it can control the not only the flow of gases, the exchange of gases, the amount of oxygen going out, the amount of carbon dioxide coming in. It also helps to prevent excessive water loss through transpiration because plants do lose water to the atmosphere through transpiration. And back to these mesophyll cells and the bundle sheath cells. Again, this is where you're going to find your chloroplast. Now, your chloroplast is what we call a double membrane bound organelle, where it has an outer membrane and an inner membrane. But equally as important is that third membrane system, and that's where you see all these green parts. And here, this is your third membrane system. Now, this membrane system, as you see, these green stacks are called grana. Each one of these grana is composed of individual discs called thylakoids. And these thylakoids are where you're going to find the chlorophyll or the green pigment. The thylakoids are connected to each other through lamella and also through the stacks. And then around the thylakoids, you have this dense liquid, and this dense liquid is the stroma. This fluid is also important because it's the site of the second reaction or the Kelvin cycle. Now, as I was stating, photosynthesis has two stages, the light dependent and the light independent. The light reaction is going to occur in the thylakoid, and the Kelvin cycle or the light independent happens in the stroma. In order for the light reaction to occur, there's two things that must be present. You need light energy, and you also need water. Now, the energy source for the light reaction is going to be the light, and the energy source for the Kelvin cycle is going to be the ATP. So where does ATP come from? We know where the light came from or where the ATP come from. ATP is going to be created as a product of the light reactions. So the water is essential because water is composed of hydrogen and oxygen. I apologize. I'm having some issues with my PowerPoint. But anyway, the oxygen, as I was saying, is going to be released from the cell as a byproduct. Oxygen is not what we wanted from the water. What we wanted was the hydrogens. Now, if you notice, I have these protons or these hydrogen ions drawn around this chloroplast. What happens is the hydrogens are going to be stripped of their electrons. And when that happens, you create a proton. When the hydrogen is stripped of its electron, the electron is going to be passed through electron transport chain, and in the linear reaction, you're going to end up with NADPH created. Now, this ATP and the NADPH are going to be generated in the stroma because they are essential for the Calvin cycle. The ATP is the energy source for the Calvin cycle, and then the hydrogens that are carried by NADPH, which I call a carrier molecule, to the Calvin cycle are going to be incorporated with carbon dioxide so that we can fix this inorganic carbon dioxide to organic sugars. Let's take a closer look at the light reactions. If my PowerPoint don't quit again. All right. So in order for the light reaction to occur, there's photosystems, and photosystems are integral proteins. They're transmembrane proteins, and these transmembrane proteins are embedded in the membrane of the thylakoid. I'll go back for a second and just remember the thylakoid here. So your thylakoid is a disc, and there's a membrane around the outside and there's space on the inside. So the photosystem is actually a part of that membrane, okay? So when we look at our photosystem, it has two parts. There's a light harvesting complex, which is this purple part, and then there is the reaction center complex, which is in the center. Embedded in the light harvesting complex is where you're going to find your pigment molecules. That's why it's called light harvesting, because pigment molecules absorb certain nanometers of light waves. Now, there's a special set of chlorophyll that's going to be embedded in the reaction center. And you're also going to have a primary electron acceptor. So again, this is your thylakoid. And you have the space, which was on the inside of the thylakoid. You have the membrane, and out here is the stroma. So keep in mind, this is a section of that flat disc. There's two photosystems, Photosystem II and Photosystem I, and each absorbs a different wavelength. PS2 absorbs 680 nanometer wavelength, and is only functional in linear electron flow. And Photosystem I absorbs 700 nanometer wavelengths and functions in both the linear and the cyclic electron flow. So let's take a look at the linear electron flow. Now there's 8 steps to this linear electron flow. And the linear electron flow starts with Photosystem II. Now, these photosystems were named for when they were discovered, not for how they react. So Photosystem II reacts before Photosystem I in the linear electron flow. So Photosystem II, remember, it's going to absorb that 680 nanometer light wavelength, right? So your pigment molecules are going to be able to absorb those photons, and they pass that energy along until it reaches the reaction center. Now, again, this is that special chlorophyll A. There's 2 types of chlorophyll, chlorophyll A and chlorophyll B. This is that chlorophyll A, and we call this one P680, because remember, what's that wavelength of light again? 680 nanometers. So, as I was saying, when the energy is passed from the pigments to P680, the electron gets excited, and when it gets excited, it jumps up to a higher level. We know when electrons get excited, they jump up a level. So when it jumps up, it is going to be accepted by a primary electron acceptor. So step 1, chlorophyll accepts, the pigment molecules accept the photons. Step 2, the energy is passed to P680, the electron gets excited, and it jumps up to a primary electron acceptor. Now what happens when something loses an electron? It becomes oxidized, right? So P680 is no longer P680, it is now P680 plus. So P680 plus is the strongest organic oxidizer that we know of. So now this is an oxidizer. So it is going to essentially steal the electrons from something else. What are the electrons going to come from? Water. So with the help of enzymes, the reaction is going to be catalyzed that splits a water molecule. And that water molecule is split into hydrogen and oxygen. Now, again, the oxygen is not wanted, so it is gotten rid of as a byproduct. The hydrogens are stripped of their electrons, and that leaves me with protons, because when I take an electron from a hydrogen, I create a proton in this sense, because all that's left is a proton. So the electron that's taken from the hydrogen is going to replace the electron that was excited from P680 and make it P680 again. So P680 is reduced, because when you receive electrons, when you gain electrons, you're reduced. If I lose electrons, I'm oxidized. So water is being oxidized, but P680 plus is going to be reduced back to P680. Now, this electron is excited. The electron that was once with P680 is excited, and it has been jumped up to a primary electron acceptor, but when something comes up, it must come down. This electron is going to essentially fall down an electron transport chain. It's going to be passed through a cytochrome complex. Now, what's the charge on the electron? Negative. What's the charge on these protons? Positive. What do opposites do? They attract. So by the energy being released as this electron is being passed down this electron transport chain, it is causing these hydrogens to create a proton gradient and cross that thylakoid membrane. This proton gradient, this crossing of the thylakoid membrane, is actually going to help ATP synthase. It actually turns the ATP synthase to stimulate the production of ATP. Now, why is the electron falling back down? Well, you can thank Photosystem I for this one. Photosystem I, of course, absorbs a different wavelength. It's 700 nanometers in wavelength of light. And so what's happening is, as this light has been absorbed by these pigment molecules and is being passed to P700, P700's electron gets excited and goes up to a primary electron acceptor. Well, what happens to P700? It becomes P700 plus. It's positively charged now. And you have a negatively charged electron here and a positively charged oxidizer here. So what's going to happen? This electron is going to be passed from the primary acceptor in photosystem 2 to the chlorophyll in photosystem 1. When that happens, P700 plus becomes P700 again. It becomes reduced. The electron that was in the primary electron acceptor is in photosystem 1 is then passed to ferredoxin. Ferredoxin then has the task of, in the linear flow, passing those electrons to another enzyme, which is NADP plus reductase. It is going to help to reduce NADP plus to NADPH. Not only does it feed the electrons into this system, but NADP plus reductase is also going to be instrumental in removing protons from the stroma, which also helps with that proton gradient. So At the end of this linear process, through the first electron transport chain between photosystem II and photosystem I, I have ATP. And between photosystem I and NADP reductase, I'm going to have a second electron transport chain driven by ferredoxin passing those electrons, and I ultimately end up with NADPH. Both ATP and NADPH will be generated in the stroma to be used in the second reaction. Now, what happens when instead of being linear electron flow, we have a cyclic electron flow? Well, first thing, we need to eliminate photosystem II. Photosystem II does not function in the cyclic flow. And since photosystem II is not a part of the cyclic flow, there's also no oxygen created. Because remember, water is split in photosystem II, not photosystem I. Photosystem I, 700 nanometer wavelengths absorbed by the pigments, past the P700. It gets excited. The electron goes up to the primary acceptor, goes from the primary acceptor to ferredoxin, but this time ferredoxin drops the electron back into the first electron transport complex. So instead of going to NADP reductase, it goes to the cytochrome complexes from the first electron transport. What was the product of the first electron transport? ATP. Because it does not go through the second transport, we don't get NADPH. So the cyclic flow only produces ATP, and it does not release oxygen. So I'm going to do a second video which will talk about the second reaction of photosynthesis. I hope this helped.